Hello, welcome back to the Art and Science Reading Group, joint hosted by Science Gallery Dublin and the Trinity Long Room Hub, located at Trinity College in Dublin, Ireland. Thank you guys so much for coming back and spending some time with us. From our quarantine caves to yours, you are very, very welcome. My name is Autumn Brown. I am a PhD researcher. Is that what we're calling ourselves now? An early career researcher at the Trinity Long Room Hub and the PhD researcher at Science Gallery Dublin. My research is on informal science learning spaces that take an art science approach and how that art science approach might transform science and change the way that we look at the culture of research and innovation. With me is Amelia McConville.
Yeah, perfect. So my name's Amelia. Hello to everybody who's tuning in. Oh, sorry, hang on. Can everyone hear me? Perfect. So my name's Amelia. Thanks so much, Autumn, for the introduction, and welcome everyone to the Art and Science Reading Group. We're so delighted to be hosting our second virtual session here tonight. Basically it's up to us to just introduce you. We're going to kick off fairly immediately, and we're very excited to have with us here tonight Anna Dumitriu, who's very kindly agreed to be our artist to interview tonight, and we're going to be speaking about her work and some of the readings that we recommended people have a look at before the session. A brief introduction of myself first. My name's Amelia, as I said, and I am a PhD student. I'm an interdisciplinary researcher working with primarily with the School of English, but also with the Institute of Neuroscience in Trinity. I'm doing interdisciplinary research on visual poetry and neurohumanities, and looking at ways to have conversations across those two disciplines. Autumn and I actually met in Trinity Long Room Hub, which we have to say a huge thank you to, as well as to Science Gallery for hosting our virtual reading group and for making it possible to have us here with reaching so many more people than we ever thought possible. So thanks again for joining us. So it's up to me now, I suppose, to introduce Anna, who we're so delighted to have with us, as I said, tonight. So Anna is a British artist who works with bioart, sculpture, installation and digital media to explore our relationship to infectious diseases, synthetic biology and robotics. She has an extensive international exhibition profile, including Ars Electronica, ZKM, the V&A Museum, Philadelphia Science Center, the Museum of Contemporary Art Taipei, Art Laboratory Berlin, and the Museum of the History of Science. She was the 2008 President of the Science and the Arts section of the British Science Association, and her work is held in several major public collections, including the Science Museum London and the Eden Project. Anna has presented talks on her work at various prestigious venues, including TEDx conferences, the Tate Modern, Princeton, Imperial College London, University of Oxford, La Musée de la Chasse et de la Nature, and UCLA. Her work is featured in many books, including BioArt, Altered Realities, which is published by Thames and Hudson in 2016. And she's written for many other significant publications across contemporary art and science, including Frieze, Art Forum International Magazine, and Leonardo Journal, as well as Nature as well. In the past, Anna's work has explored synthetic biology. She's also worked with the MRG Grammar Project to create artworks using CRISPR gene editing technology, and also collaborated with Beyond Seek at the University of Birmingham to explore the biochemistry of DNA. Most recently, she was also featured as the answer to a question on the popular BBC Two series QI. So Anna, thank you so much for being with us here tonight. And I'm going to turn over to Autumn in just one second to ask our first question to start off our discussion. But also just to let our participants know, and our audience that's joined us here tonight, that we're hoping that people can participate as much as possible in our conversation this evening. So the way we're going to run it is we'll have a general Q&A with myself, Autumn and Anna are going to have a conversation about Anna's work and about some of the reading. We would encourage everybody to ask us questions, share your ideas, your provocations, your comments as you're listening to this. And then at about 7 o'clock, we're going to kick off with our proper discussion and take your questions and ask them to Anna, and hopefully facilitate as much of a conversation with everyone as possible. So thank you so much, and I'll pass over to Autumn now.
Thanks, Amelia. Yes, this is definitely an informal conversation group. The original iteration of this was in person in Science Gallery Cafe. So do throw those comments, those questions in there, and we'll be really, really excited to share them with Anna. But first, Anna, would you mind describing the plague dress that was mentioned in our reading? If you all didn't get a chance to do the reading, Don't worry.
Yeah, so my plague dress is a 1665 dress design with a long gown from the plague era, Restoration era dress, which is dyed with walnut husks, and it's got an embroidery across the front, which is original period embroidery that I managed to acquire, and then impregnated into that embroidery is the DNA from Yersinia pestis plague, which I managed to extract in the lab at the National Collection of Type Cultures in London. And then when the dress is shown, it's surrounded by lavender and stuffed full of lavender.
Amazing. And what is the significance of the walnut dye and the lavender? Was that?
Yeah, the dye was used by Nicholas Culpeper, I believe, Culpeper. And the lavender was what they used to stuff into the nose, kind of the masks of the plague doctors in those days. So yeah, you're looking at it there.
Beautiful.
So yeah, it was just stuff full, and actually the middle bit of the they have this thing called a bum roll, basically, that goes around the waist. I think that's where the name comes from. I've never really managed to find the history of it, but this is stuffed full of turmeric and other spices that we used as treatments, and kind of the spices that used to stuff into the plague doctors' masks and things at the plague era. So it kind of, it's a very olfactory experience when you actually see them.
Amazing. And were any of these treatments, either the walnut or the lavender, actually effective in preventing? Okay. So they were just sort of folk tales.
It's actually very, very deadly plague. I mean, it's one of the biggest killers of, if not the biggest, one of the biggest killers of humanity. And as I say in the article, one of the most sublime organisms from history. So it takes quite a lot of treatment. And I mean, even now without treatment, you're looking at a 50% death rate.
Goodness.
It's quite a dangerous kind of illness.
And I noticed you mentioned there a keyword, the sublime, which for anybody who had a look at Anna's article in Antennae magazine, you talk about this wonderful concept of the bacterial sublime, and there's actually, like, totally by chance, there's actually a link to famous Trinity alumnus Edmund Burke. So I wonder maybe you could tell us a little bit about what is the bacterial sublime and how do you conceive of that notion?
Yeah, well, I was fascinated when I first came upon the notion of the sublime. It was quite a few years, a very long time ago actually, and I was stunned. I was like, yes, that's the thing I'm interested in. That's it. But and I'm interested in these bacteria, and that's what's going on in my head. So then I started to go through all this research, which seemed which it became a sort of ridiculous rabbit hole, actually. I started to do a PhD, which then I ended up finding that it didn't really fit with my art practice to be working in that way. So I was kind of exploring all these things. But this idea of the sublime that Burke talks about is really fascinating, because he actually mentions a few things. He creates a list of these things which he considers to be sublime, which is kind of odd. So he found there was this original text that was republished and became very popular around that time, Perihipsu, I'm probably mispronouncing it, by Longinus, probably mispronouncing that as well. And this was published, and there was an article in the Spectator around that time, and Burke then took it upon himself to write this philosophical inquiry, which I have here, the philosophical inquiry on the beautiful and the sublime, where he kind of makes a list. Look, you can see all my notes in it. He makes a list of all the things which he considers to be sublime, and he talks about that. And you have to understand that he was writing this in the sort of early 18th century, so they had seen bacteria, animalcules, as Van Leeuwenhoek called them. But so they'd been observed by that time. So he kind of had a sense of this, and he mentions in the text that the very tiniest things might be sublime. So, and then he also mentions the obscurity, not being able to really see something or make something out is an aspect of this idea of the sublime in Burke. And I've got here, because we were talking about this the other day. This is a replica of Van Leeuwenhoek's microscope that he used to look at bacteria under the microscope. And you see, you put your sample. I've kind of used a bit of sellotape here. This is a piece of Irish linen, actually. You put your sample there, and then you hold it up basically to the light and look through, and then you can sort of see through this tiny hole. You can see the whatever it was, and so that's what they were using to look at the kind of the first bacteria that was seen. And so he was aware of that when he was writing this. So that's what really fascinates me. And then obviously Immanuel Kant, this fella I struggled with, you can see all my notes in here. He talks about the sublime in the sense that it's not something that you can put in a list, really. It's not, it's not a mountain or a big dark hill or a tidal wave or something like that, which Burke might talk about. Kant says it's all in the mind of the person perceiving it. So it's that sense, it's that relational thing then that he talks about. So I kind of, and this relationship to this idea of the sublime, and the fact that, I mean, particularly plague, and we're living through another pandemic, not nearly as at the moment anyway, as dramatic as the Black Death or something like that, but it changes the world. I mean, it stops everything. This is what I was always talking about in terms of how much plague has changed the world, and it killed half the population of China, third of the population of Venice were killed at one point, third of the population of London roughly were killed. I mean, we don't know the exact figures, but and it brought in things like public health. It created huge changes to how we live our lives, governance and trade, and all these things, and we're kind of living through that again.
Absolutely.
It's really interesting that you say too, that the information that we have from that time is largely from diarists like very, very famously and mentioned in your article, Samuel Pepys. So much of what we know about what happened, the fire of London and the plague that struck, were from diaries. And it's extraordinary too. I think perhaps you've seen online and in different places now that everybody thinks they're the new Samuel Pepys, which I think is kind of extraordinary. So it's really interesting to imagine what art might be produced or what we might learn going forward from the diaries that are being kept now. And it's interesting too, your work, I particularly love it, and it resonates with me because there's a shame. I love science, but we have this problem of modernity. We are always looking forward and so rarely looking back at history. Actually, citation patterns, I have this nice little quote pulled out. Citation patterns show that the older research is considered less relevant than more recent research. This is called a research front. Science really does pay little attention to its past, sticking mostly to current work. And then that the present-centric kind of view of things allows scientists to keep looking forward without really often taking lessons from looking back. And I think your work really, really highlights that we call this the new normal, we call this kind of unprecedented, but it really isn't.
No, it's not.
It really, really isn't.
No, most of it, apart from the tech side of it, most of it's happened previously, and the tech stuff's really just stuff that was happening in different kind of ways, in the papers. They used to publish papers about, several times a day, so there'd be this thing being handed out all the time. So the news was really quite up to date, probably more than we have to an extent now. So it's interesting because we think we've really moved on We think we're so different from like 350 years ago, Samuel Pepys, but we're not. That's what I love about reading his diaries, is that it's like, you know, these people, they're just normal people. He's mainly like going to a coffee shop, worrying about where he's going to get his clothes, worrying about how he's going to visit his girlfriend. That's quite a ongoing theme in the book. Putting up with his wife, you know, these sort of things. It's just, it's a kind of fascinating, it's just so fascinating. And yeah.
Perhaps Anna, linking the lovely quote that Autumn shared with us just there, and your comment previously there where you mentioned linen, and I know in some of our preliminary conversations before this event we talked a lot about linen, and I think maybe, well, I'd love for you to talk a little bit about linen, that I suppose that something that links not just disciplines but I suppose generations and centuries. You made a really interesting point about linen before, and I'd love for you to elaborate a little bit on that.
Well, linen's so fascinating because it's this first bio-textile, if you like. It's made, I'm sure people in Ireland know this, through a process called retting, which is a kind of where the flax stems are put in these stagnant pools and rotted away by bacteria. So it used to be quite a smelly process. It's still made in that way, but they do it in tanks now rather than the traditional way of having these kind of pools. And it reappears, there's these sort of themes that reappear in textiles and in medicine and in science throughout history that I'm fascinated in. Linen's one of them. And I think I was talking about, so Van Leeuwenhoek, with his microscope, the reason I've got a bit of linen here, Van Leeuwenhoek knew how to grind these lenses because he was looking at the thread count, because he was a linen draper. And so it was the linen that kind of came in there. And he saw Robert Hooke's Micrographia book, which is famous drawing of a flea in it, which was published in, I think, 1665 as well. Not knowing, he didn't know that the flea was this big vector for the plague to be transmitted. But Van Leeuwenhoek was less interested in that, but he saw there was a lovely drawing of the threads under the microscope. So Van Leeuwenhoek was inspired to kind of look and do his own lens grinding. And people, I mean, I think they sort of think they know how he did it now, but it was in question until very, very recently, how he achieved these fine lenses. And then you've got this whole thing with linen being the fabric that you could wash. So silk was never washed. It's too fragile. I mean, when the sort of the old washing methods was to put it in a copper and boil it up or something like that, so beat it with a rock down by the river, or use mangles and that sort of thing. You can't do that with silk, it'll tear it. And you can't do it with wool. Wool's kind of antimicrobial actually, so doesn't necessarily need it. You can hang it outside and you can clean off the stains, and so wool's good in that way. But linen you can boil up, and you can you know, bleach it in the sun and things like that. So because of this thing that you could wash it, linen became a representation of cleanliness. So the piece in the article that I mentioned is called Clean Linen, and it's about this fact that clean linen was considered proper cleanliness. So you didn't wash your body. Going out and having a bath, that'd be weird. You'd catch a cold if you did that. But if you were wearing clean white linen, that would meant you were a clean person. When I say clean, it could be 3 weeks old clean linen, but that's quite clean for those days. And so there was this idea of cleanliness, that if you washed the linen would regulate the humours in the body, and it would leach these toxins out of the body. So the dirt would go out into the linen, and then you'd wash the linen. So the piece I made with that clean linen, I think you've probably got a picture of that one, haven't you, Amelia?
Yeah, I get it, you know.
The piece I made there, it's embroidered. It's an original French linen nightgown from the period that it's prior to the end of the time of humoral medicine. So it's an antique gown that I've then embroidered with white work embroidery in various parts of it. So around the neck, you've got diphtheria bacteria. You've got Mycobacterium tuberculosis there, Yersinia pestis there, and you've got the bacteria that cause scarlet fever, which you can't see, they're a bit further down on the stomach. And then in those areas, so you've got exactly where they'd kind of be in the body. In those areas, I've actually impregnated them with the DNA of those 4 kind of really significant organisms of the time. And I should say that DNA is sterile, it's not infectious. So you can work with plague DNA. We extracted it from killed plague in the lab. I have actually handled and worked with live plague in the lab. But when we do the DNA extraction, in order to be able to take it out of the lab, it's killed plague, and then we extract the DNA, and then it's been through rigorous kill protocols and things like that, working with Public Health England at the National Collection of Type Cultures, where I'm their artist in residence.
Amazing. Actually, that was one of the things that I really wanted to ask you about. As an artist who's frequently working with scientists in laboratories, what is that process like? But also, why do you do it? Why is that important to your artistic practice? So many artists don't go near science, is not their thing, doesn't really inspire them. So I suppose, what is it that led you to work with scientists and in laboratories, and what has that been like?
I think I was really fascinated in microbiology, and it's important for me to do stuff hands-on. I can't explain to people or tell the story or understand it without actually having done it myself. So I need to kind of absorb everything and learn as much as I can in order to be able to communicate the story. So if somebody wanted to kind of go into the details of how I extracted the DNA and stuff like that, if I ever got questions on it, I would know, and I have all the proofs anyway, but still I can explain everything, and that's really important. Also, in terms of kind of, I did a piece called Confronting the Bacterial Sublime, which was a written piece that I did in response to going into the Category 3 lab and handling plague bacteria and handling anthrax as well. But mainly it was about writing about being in, working with plague. And that's kind of interesting, because when you're doing it, you can't really experience the bacterial sublime. You're there with it. You have your hands in these glove boxes you've probably seen in movies. But what you don't know is they're kind of like inflated. So because you're under all this negative pressure, you're double gloved, and then you take your gloves to the lab coat, and then you put your hands into this thing, and then you're handling the bacteria. And then they say things like, use those scissors there to cut open those Petri dishes, so you can do some work in there. But you have to actually, in with these big inflatable gloves, you're actually kind of cutting with scissors and being very careful not to cut a hole in your gloves. Obviously, I mean, it's all under this negative pressure, so it's being sucked away from you. You can't actually catch it from just touching it, but still, there is a sense of this to an extent. But actually, you're more worried about, in my case, I was more worried about doing it wrong and looking like I was an artist in the lab rather than a microbiologist, because I've been, not that I am a microbiologist, but I'm fully an artist. But I wanted to, I kind of wanted to be accepted as a microbiologist, because I've done many years of this. So I kind of felt that I needed to do that in the lab, which is an interesting kind of thing. So I wrote a reflective piece on that experience. And one artist, I think Hero Riley, said to me that she said, oh, that's interesting, because basically the procedures of the lab took over and you were following that rather than being able to have this artistic thing. So it's interesting how these things kind of impose on each other, I guess.
Well, I'd love to ask you though, Anna, like, on that very topic, you know, we obviously, one of the things that we're very interested in breaking down through the reading group is this, you know, totally binary opposition between arts and science. You know, we just don't believe that binary should exist. But your work, because you, as you say, like, you have extensive experience with actually microbiology and working in lab-based scenarios and everything. But I suppose in terms of the artistic impulse for you and the interest in science and that mutual influence, I mean, do you use the science as an influence? Does artistic inspiration strike when you're engaged in learning about the science or reading about it, or how does that relationship work?
I mean, definitely. It's the like the research, looking into the history, and I always talk about the boring moments in the lab. So when you're doing work in the lab, there's quite a few boring moments. I don't know if people listening in on this have done this. There's bits where you have to run PCR machines, and so it's like, we've got to put it on for 5 minutes. And then he's just standing by a machine that's spinning things for 5 minutes, and we've got to put it on for another 5 minutes. And we have to cool this down for 20 minutes because you have these protocols that tell you what you're doing here and what's next and what's next. And so there's quite a lot of boring steps in the lab. And it's during those moments when I get to talk to the researchers about any old rubbish. And that's where a lot of the really fascinating stuff kind of comes from, I think. So that's really good. And I've been working with my longest collaborator now for well over 15 years, pushing 20 years now. So we have like quite an interesting kind of relationship of ideas, and he knows how it sort of turns when we're going to start doing something. So that's interesting. He's retired now, but he's writing a natural history of bacteria at the moment. So as a written book, but he'll be able to go back into the lab. We actually had some very lovely plans to do new work together in the lab with the strains of bacteria from the National Collection of Type Cultures. That's all kind of got delayed because of the pandemic, but we're going to do it afterwards.
Absolutely. And actually, so on the kind of art science collaboration and art science approaches, one of the other Edmund Burke quotes that you've included in your article, which really, really jumped out to me, is he wrote, "When we go but one step beyond the immediate sensible qualities of things, we go out of our depth." And it is really interesting to me at those spaces when we feel out of our depth, is it then that we should employ art science approaches? Is it then that we should experiment with different forms of inquiry or different forms of making knowledge and understanding the world?
I would say probably yes. It's kind of as simple as that. I mean, in a way, I don't think we can kind of help it, is we have to find other ways to understand things and talk about things. It's with things like the pandemic at the moment, there's so much that we don't know still, is that how can we kind of help people who are in a space where we're going, well, what should I do? What does this mean? You know, what's next? Or my dad, for instance, said, when's this pandemic? When's it going to all end, the lockdown? And my mum, and he was asking my mum, and she said, well, if I knew that, I'd be on the TV, wouldn't I, instead of those people? So I quite enjoyed her perspective on that, because it's like that, but people just ask and they expect someone to know. And I know there are scientists out there, but we've never seen a pandemic. We've never seen this organism before, and we don't know exactly when it's going to be safe to fully go outside and these things. We don't know what's going to happen with the vaccine fully. There's a lot of hope, I think. Of course. But yeah, we don't know exactly the timeline, and there's a lot of uncertainty.
Absolutely. And Anna, on that, I know it's quite a big question to ask, but especially considering that your work in recent years and with a special focus on plague dress, you know, again, that work we've been discussing from 2018, I wonder, do you see, as it is in the moment, I know, as we said, the situation is still evolving, but do you see the potential for there to be, I suppose, any knowledge gleaned from this kind of work, these kind of art science approaches to different artistic practices and exhibitions? Like, do you see them having relevance, if not influence, on how we respond to the current pandemic, or do you feel that it's maybe still evolving in a very sort of new space?
it's still evolving, but I mean, that doesn't stop art from responding throughout, and I guess those responses will change. I've been making, I've got this piece called Xvoto, and it's a I think you've got an image of it, actually.
Yeah, I got it up for you now.
Of it. And it's a piece where I made these kind of, they're like basically secular votives that you might see hung into like a Greek church or something like that, but each one is representing something about infectious diseases. So it's either somebody taking antibiotics, or somebody who works with antibiotics, so somebody had an infection with an infectious disease. And the main focus of it has been antibiotic resistance. And you can see these ribbons that are there, and they're all dyed with different things to do with infectious diseases. So the orange ribbon is Prontosil, a very early antibiotic. The yellow is turmeric. The red is madder root, which was used as an antimicrobial dye. Woad as well, which used to be, which is antimicrobial, but used to be used, painted on the faces of the bodies of the warriors, the Scottish kind of warriors, like in the film Braveheart or something like that. And the people think that maybe the warriors that painted their faces and their bodies blue might have survived better in battle, because if they get cut, they wouldn't have necessarily died of infection. And then some of these other ribbons are dyed with different, like genetically modified streptomyces and the Staphylococcus aureus from my body and things like that. And then, yeah, you were just going to show, I think, the image, some of the starting images that I've been kind of creating. I've got a whole box full now. I've been doing one every day, kind of inspired by, you know, things that happen on my friends' Facebook posts or something I've seen. And so, you know, these are very basic ones. We've got a face mask and a lung. And then up the top there, that's the Starlink satellites. The Elon Musk's launched these weird satellites that are going across the earth in these, well, you see them on Facebook. I've never seen one. I'm really dying to see them going over, but apparently they come over in a line and it looks really weird. And I thought, well, and I grew up in Hastings, where famously we had the Battle of Hastings, where Britain fell to the Normans. And in the Bayeux Tapestry, there's the Halley's Comet flying over is embroidered into it. So I was sort of thinking about this Starlink as this kind of technological comet flying over us as this sort of portent of this time, much like the Battle of Hastings and Halley's Comet. So I was sort of, there's lots of things. And then I've got like my friend walking, I've got one friend who's doing COVID-19 testing in a hospital. He posted on Facebook a picture of his nice walk to work. He was like, this is the nice bit before I get into the lab when I'm doing my commute. And he had put a photograph of some trees and a pretty sort of woodland. So I drew that, and that's, you know, I'm going to kind of, that's one of his stories, one of his memories. So there's all these different things emerging. So I'm kind of like, if people have got things that they think should be included, I'm kind of collecting things to include in that and hope it will grow and grow. So I'm probably going to dye the ribbons with turmeric, because I read that in India now all the turmeric root is sold out, because they're using that as believing that's a treatment for COVID-19. They think it's helpful for that. It's probably not that helpful, but but I like the idea that it's being used. Sometimes I use things, I don't necessarily think these dyes are antimicrobial or particularly antimicrobial. Turmeric's a little bit antimicrobial, but not enough to work as a kind of antibiotic. It doesn't have high enough levels of curcumin, the active ingredient, in it to affect us when we eat it. It works a bit in a petri dish, or if you're washing things with it, it can be a little bit like, it could replace a disinfectant to an extent. So I'm probably going to kind of dye these ribbons with the turmeric or anything else I kind of find like that, and then hang these things on it. And the NCTC is related, well, they have lots of different collections, the National Culture Collection, so they do have a national virus collection. I'm not saying I'm going to impregnate it with COVID-19, but it could be possible to take a small section of out of frame DNA, or RNA, I mean, of the organism, and use that, or just to create a small section that from the genome that isn't the organism, which anyway isn't a problem once it's got rid of its lipid membrane. But that's the sort of debates I go into with I wrote this book, Trust Me, I'm an Artist, which I actually did an event for at the Science Gallery Dublin, and that's the sort of question I go into there, because I'm kind of fascinated in that. What can I do? What do I need to do to make it safe? And then there's this frisson of this, and I think that's sort of the sublime, is this knowing it's there, even though it's safe. And that's somebody that, like other writers have talked about, this idea of actually the sublime isn't terror. It's got that safety. So it's like you're seeing a tidal wave, but you're seeing it from a safe position, or you're watching a volcano. But if you're actually on the volcano with the lava coming towards you, it's not sublime anymore. It's just terror.
I think
so it's got this removal.
I think it's really interesting too, and also it almost is like what it feels like to be on a boat and look out at open water, just completely overwhelmed by the immensity of it. I think almost the way that you described the sublime in your work, and the sublime from the very limited reading that I've done of Edmund Burke's kind of interpretation of it, is that it's this feeling of being so finite and fragile yourself when you are faced with something so great, immense, or powerful and tiny, and have that feeling of like overwhelmed, and potentially maybe something related to awe or humility. Maybe
definitely. No, it's to do with that. It's to do with this kind of great leveler thing, I think, as well, is that rich, poor, whatever, we're all equal in front of whatever these sublime things are, like COVID-19. However rich you are, however powerful you are, you might still end up on a respirator, or at least getting oxygen in intensive care, as we've seen. So actually I should say Burke originally kind of had his reflections on the sublime, the initial ones, where there was a flood in Dublin that set him off on all this thinking about it, apparently. So floods are quite important.
Fantastic. I think on that note, Anna, we'll definitely start opening up to our questions from our attendees, and the questions have been flooding in by the Q&A section. So please do get your question in now if you want us to try and address it. Hopefully we'll get a chance to touch on at least everything that people are bringing up. Maybe just coming on from what yourself and Autumn were both just saying there, Anna. There's one or two questions that have come in asking about the first of all, the element of risk. That question comes from Camilla Fanning, who is an Irish etching artist. So Camilla asks about how much risk is an element of interest in the work. And then there's also a question from Dion Ellis Taylor, who asks, is there an element of thrill seeking in handling these almost dangerous animals? So talking about those dangerous bacteria and DNA. I know you were talking there about the idea of the plague being dead, but is that an element of your work?
Not the thrill seeking. It has been suggested. It was suggested at the Science Gallery Dublin, actually. We did an event for my Trust Me, I'm an Artist project, where I was talking about the idea of building a category 2 in a lab, and it was suggested that I was interested in working with more and more dangerous bacteria from that perspective. But I don't think it is that perspective. It's their significance to humanity. And there aren't many artists working with infectious diseases, because one, it's quite hard. You have to go through enormous processes to be able to do it and kind of dedicate your life to doing it. But I don't think any microbiologists would like, I think I'm not a microbiologist, but I would sort of see myself as in the same sense as microbiologists. And I don't think that they do it from the element of thrill seeking, because as the other question, the risk thing, the risk's really quite low if you understand how to do it right. Like I'm not like going into a category 3 lab to handle plague just to get this thrill from it. I'm not. If anything, I was trying to do it right, like science-y right, which is different. But I think it's the history of the organism that is interesting to me. So whether it's the Yersinia pestis, which causes plague, or MRSA, which causes skin and whatever infections, blood infections, that's kind of superbug, or Clostridium difficile. I mean, is that a thrill-seeking bacteria? It causes ulcerative diarrhea, and it smells quite bad, and it creates biofilms, and I've made artworks. My spindle piece is on my website that you can see under the From the Field section, and that's made with these Clostridium difficile biofilms. Clostridium means spindle, and the sort of the early Clostridia, like Clostridium pastorinum, which was originally called Clostridium pasteurianum, was discovered by Sergei Vinogradsky in his explorations of the retting of flax. He was this pioneering microbiologist who looked at whole ecosystems. So I wouldn't say there was an element. Oh yeah, you've got it.
Yeah, I have it there for you.
So you've got my Clostridium difficile biofilm pendulum kind of bits on this piece. Well, they're like spindles, really, drop spindles. And so I would say no to the thrill seeking. I would say, in terms of the risk stuff, what's interesting is your questioner was an etcher, you said.
Yes.
Yes. So I always compare what I do to the feeling of etching. Actually, it's very similar. Working in the microbiology lab, you're working with these things. They're not going to leap out and get you. I don't work that much with viruses. So we have a different perspective here on, because the world thinks about all these things flying around in the air or whatever at the moment. So I'm not working with them. I'm working with these things that are, in a Petri dish. Mm-hmm. You don't pick them up and sniff them unless you know what's in the Petri dish. That's very bad. There's some things you can actually catch by sniffing a Petri dish, but they don't tend to leap up and kind of get you. You have to be careful, but and if you're aerosolizing them and doing those sorts of procedures and stuff, you have to be careful, and you have to follow all the protocols in the lab. And I can't tell you how much health and safety training I've done in my life, but etching, it really feels like microbiology. There's sort of working with these acids, and do you put your hand in, do you not, what gloves do you need to wear, and you've got these big sort of cabinets with the extraction, and you have to go through every 5 minutes, you need to feather the etching plate or something. I've done quite a bit of etching in my time as well, so I have a sense, and there's a lot of processes that come from things like gram staining, which is where you put the dyes on the bacteria to dye the cell wall to be able to look at them under the microscope. A lot of them are quite artistic processes. It actually feels very much like making art.
Amazing. So we've got an audience question and an audience comment. So question from Andrea Bendelli: if you would be able to say a few words about the antibiotic resistant quilt, and that he loves this work, not only because it was commissioned by Science Gallery. And we've got a comment from Sahar Ahmed. We grew up drinking turmeric and milk, now being appropriated as turmeric lattes for being anti-inflammatory in Pakistan and India. So that's really, really interesting to hear about your work with turmeric. And she says, thank you.
So the antibiotic resistance quilt. So that was a quilt that I made, commissioned for one of the shows at the Science Gallery Dublin. It was shown there, and the piece is impregnated with silk squares that I grew in Petri dishes with the WHO's top 10 superbugs, because antibiotic resistance is this massive existential threat for humanity, which the UK, the England chief medical officer previous to Chris Whitty, who's always on the TV now, but Dame Sally Davis said that it was as big a threat to humanity as climate change. So that's quite bad. And it's just because the bacteria that we use to treat diseases are slowly evolving, or sometimes quickly evolving, resistance to the drugs that we use to treat them. They've always been doing this in their own way in the soil or whatever as they evolve, but now it's much speeded up and it's targeted to those drugs that we use to treat them. And So we need to be finding solutions for this, understanding the genomics of the bacteria and things like this. And the COVID-19 crisis is a big problem for this, because there's a lot of use of antibiotics now in the bacteremia, lung infections, particularly bacterial pneumonias that people are getting, and they're being given prophylactic antibiotics for, if they're going to have a ventilator or something like that. Also if like dentists are now giving people antibiotics for toothache, rather than having them come into the surgery, because they can't do that. So to keep them at home, keep them okay, they're being given antibiotics. So we're gone back to using a lot more antibiotics than we were, and we were trying to really reduce that, but now it's gone back up. And one of my collaborators said it was a bit like throwing a bomb at the issue of antibiotic resistance, this pandemic. And we're actually doing some more work on this together. So she managed to get some funding. It's Jane Freeman, who I made Spindle with, and some of the other work with Clostridium difficile with. And she's managed to get some funding before this to work with me on on some more work. And we decided that we're going to take some of that focus and put it into this issue of antibiotic resistance. So it's this soft sort of cozy quilt, the antibiotic resistance quilt, but it's impregnated with the top 10 WHO superbugs resistant organisms, things like, well, it's got a drug resistant E. coli, and there's some super gonorrhea, things like that, basically. They're really nasty, problematic ones, but they're all sterilized. And it was kind of an offshoot, because I was working on another project at the same time for the Science Museum in London for their show Superbugs. And what we wanted to do was create a series of Petri dishes which you could show in the gallery that were the superbugs, but were sterile. And so we were growing lots of these for that, and trying to do experiments to make things that one of my collaborators described them as basically stuffed. So they look alive, but they're dead. And that was complete trial, that project. But as a sort of nice, fun project on the side of it, much more simple to do, was to actually impregnate these bacteria in some of the Petri dishes whilst I was doing it, and having all these top 10 superbugs there. And I could make these two projects at the same time.
It sounds amazing. And awful. And the other project sounds a lot like almost bacterial taxidermy, is essentially what it feels like you've described.
It was a nightmare, actually. It's really hard to do. I had a lot of collaborators on the project. I mean, it was really effective in the end, and it looked really good in the space. But I for the amount of nightmare it was, I really under budgeted how much I should charge.
Anna, we have a question who's come in from that's come in from Radek Predspelski. Radek has actually brought up the other bioartist, Eduardo Kak, who I know I think Kak's work is amazing, and I think he actually features in that edition of Antennae you sent us. There was a feature on some of his work as well. So Radek asks, Eduardo Kak said bioart is in vivo, which for me suggests a certain metaphysics of presence, which can be seen in contemporary art's utopia of liveness, i.e. bioart, performance art, etc. It seems to me that your work problematizes Kak because your notion of bacterial sublime weaves together material things and immaterial concepts, a bit like Karen Barad's idea of mattering, i.e. the processes of matters mattering. How do you agree with that assessment?
I decided I would take a broader definition of bioart, which means art made somehow with living things. But in terms of pathogenic infectious diseases, it's very difficult to have them alive. So you can't do that. But also once you accept that you can kill them, and exhibit them, then it makes your life a lot easier in terms of getting the art out to audiences, and that's something that's very important to me. And the fact that they were there, there's this trace of them, there's this history of them. To me, that with the sort of sense of the sublime thing, these invisible organisms, that still gives that to me. I hope it still gives that sense to audiences as well. So that's how I respond to it. I have talked to Eduardo Katz about this, and he's very happy with my repurposing of the term. I think he likes my work, he told me so. But obviously, you know, he's a great pioneer. He coined the term, and his work is amazing. I was reading about that when I was a student, and you know, it's a huge inspiration to me. So I've talked to him a few times about all this stuff. So he is a really great pioneer.
Amazing. That's really cool. It's also nice that you all have had the opportunity to chat about these things too. We've got another question. It's from Fiona Johnston. She says, is Anna able to say anything about the COVID and Domestic Violence Commission that she is working on for art data and health?
Yeah. So just before the pandemic started, I was commissioned by Art Data Health at the University of Brighton to work with a domestic violence charity called Rise in Brighton, and to work with health data in collaboration with them. And the pandemic started. I was about to do a few days of workshops, and it literally was, I think, it was the week of about the 15th of March or something around then. So it was just before the lockdown official, but the universities in the UK had already started to close. And so all the workshops were cancelled, and it wasn't really safe to go ahead. And so that caused a lot of rethinking. I want to do the project, but also there's this really important issue of women are at home and they're locked in with partners who may be abusive, or not just women, but particularly women and children being locked in at home with partners who may be abusive in some form or other. And I was also thinking a lot about how, where I live in Brighton, just up the road from me, talking of writers, Virginia Woolf lived in Rodmell. And she wrote this very famous text called A Room of One's Own, where she said, if you want to be a writer, you need £500 and a room of your own with a lock on the door. And to paraphrase her expansive and wonderful book or essay. And I was thinking about this idea of the room of your own and how you could create a safe space of your own, but these women don't have it. And then I was reflecting on the images of the temporary hospitals that were coming up, particularly the ones being built that I'd seen being built in Wuhan and in China. And you know, it's the opposite of a room of one's own. So what I did was I created an online workshop, a kind of web workshop, which was a practical workshop about playing with the idea of a room of your own, which could be talking about the themes in domestic violence. So this was to work with the charity team. And then the rest of the work that I've been doing, this kind of sort of the opposite of a room of one's own, to make explicit this issue. And that is, it's representative of one of these temporary hospitals. So I'm currently in the process of making lots and lots of tiny little hospital beds, which is what the installation will be, based on these temporary hospitals. And so they'll sort of stretch out as a kind of large, but I think they're about this big each. So there'll be like a floor area, or I think they'll go on a large table in the installation space, that if that goes to plan. And then with little mattresses and little pillows and things like that. And then there'll be embroidery on them, and that will be focused around the data to do with the situation with women. So I'll be looking for, I've been collecting a lot of information about the data from the press. I'm also going to be talking to the people at the University of Brighton who I'm working with on the project, and with Rise, hopefully, to get some input about what data that I should represent in this form. And I want to do that by embroidering it onto the kind of the bed coverings.
Amazing. Yeah, it's super to hear that that's how you're going to move, I suppose, move forward with your next piece, and how it will relate to the current, I suppose, the current situation we're in. I suppose that kind of links a little bit to a question that came in from Daniel Scarantino, who asks, have you done or would you be interested in working with modern fabrics, particularly medical grade fabric or PPE, like masks or gowns? And I know, Anna, you might have sort of already answered your question about how you're relating your work to the current crisis, but yeah, is that something you would consider moving forward?
I have embroidered a lab coat, which is a very early work. I did it, I think, about 2006. It is on my website under, I think, Normal Flora, under the tab on my website, Normal Flora. And it's a white work embroidered lab coat. And I think we had a little, when we were talking originally, preparing for this, we were talking about this book. I think it's a reading group. I felt the need to bring out books that inspired me, and this one, The Subversive Stitch by Rajica Parker, was a huge inspiration for me. And in this book she talks about how the role of women, basically she talks about how when natural science or natural philosophers were first doing this, like men were doing science and trying to work out things like the age of the earth or the weight of the earth or something like that, the sort of the highest level of achievement for a woman was proficiency in white work embroidery, which she should do by candlelight and hunched over wearing a corset and stuff like that, and the stitches are microscopic when you look at white work. So I've been playing with this idea of embroidery and kind of doing science through the lens of embroidery, inspired by what I read in that book many years ago, a very long time ago. It's been a big inspiration for me. But that piece is I guess using something like that. I did a sort of performance thing, but we never did anything with it, with one of my former students at Brighton and Sussex Medical School, who's now an obstetrician in Leeds. So she's actually working in an operating theatre with women who are having babies who might have been exposed to COVID. So she sent me, I've done quite a few votives about her PPE, drawings of her PPE and what they call PPE face, you know, this sort of where been wearing the mask and stuff. So she sent me quite a few of those. We did a sort of performance about putting gloves on in a sterile way that she would have to do to put them on before she goes into theatre, because normally what I do is, like, when I put the gloves on, it doesn't matter if I get stuff on them, really, because either I spray them with alcohol if you're going to do some like synthetic biology, or you're basically trying to stop the stuff from touching you rather than you getting the stuff on it. So so that was a very different thing. So we sort of did that, but we did it in a kind of performative way. We made a video, but I don't know if they ever saw the light of day. Actually, it was one of those sort of playful things that you do. It's possible that I might. I haven't got anything planned at the moment, but it's possible.
Amazing. So we do have another audience quote, and then another or comment, I should say, and then another question. So from Claire Moriarty, she wanted to say, what a beautiful talk, interweaving art, science, and social consciousness. Really inspiring to see where art practice can take you. And then we have another question. This is from Lyles Gaines. I was interested in the section of the article related to TB. How do you approach it as both historical epidemic and a current one? And how are our perceptions of it have changed over time?
Well, yeah, I should say, so TB is still the world's largest infectious killer, kills about 1.5 million people a year. We have a vaccine, but it's not that effective. We have drugs to cure it, which do cure it. It is curable. So if you're treated with drugs properly and effectively, then you have a very, very good chance of not dying from it. But a lot of people still die from it because they don't have access to drugs, and there's huge amounts of stigma in the world now on this. And that's something I'm very interested in, is this issue of stigma around diseases, and also around domestic violence, talking about that as well. You know, these things that you just shouldn't talk about. And so there's that side. So in the past, we used to talk about it as the sort of, it was sort of as a romantic disease in a way. It's been with us since, you know, since as long as any disease pretty much. Leprosy or Hansen's disease is the oldest disease known to man, but they sort of come around the same time. They found TB in Pliocene era mammoth bones, I believe. So it's been with us a long time, and it's been the biggest challenge for microbiology. It's really clever and slow growing, and we still don't fully understand everything it's doing. So there's that side. I went to Tashkent last year with the Médecins Sans Frontières conference on tuberculosis in Eastern Europe and Central Asia, and I gave a talk on my work, and I exhibited my artwork around TB there. And that was a real eye-opener. I mean, that was fascinating because you had scientists from all these different countries and scientists and healthcare workers and all sorts of people in the field from sorts of countries that almost never get to see the kind of work I would do. So you had people from Afghanistan and Kyrgyzstan and Uzbekistan, obviously, and India, but lots of different countries coming there. And so that was interesting because it had a really good reception to the work. And there was they were talking about, actually, the health minister of Uzbekistan said in her summing up at the end of the conference that people should think about collaborating with artists to get their stories out to people and to kind of try and break down the stigma. But you have to wonder whether these stories, these myths we created about the poets and this beautiful death and this sort of stuff was kind of a kind of manipulation of the idea of stigma. Because rather than thinking of people as poverty, you know, being in a poverty situation, you could think of them as highly creative poets wasting away or something like this. And, you know, Lord Byron said, I think I should like to die of a consumption, and then all those ladies would say, oh, look at that poor Byron, how interesting he looks in dying. And so it's this term of like the idea of being pale and interesting. It actually no, that's what it comes from.
I was just taken by surprise. It was great.
It's also and I'm working on a project. Another thing I'm doing now during the lockdown, I'm working on a project called Susceptible, which is commissioned by Driver Arts Driver, and it's in collaboration with the Cryptic project led by the University of Oxford, looking at their new research. So I've got all the data from their latest publication, which they published with the press release, groundbreaking publication. And I think you've noticed that scientists are very reserved in using terms like groundbreaking publication. So the fact that this made it onto the Oxford University website with the phrase groundbreaking publication, it's really groundbreaking. And what they've managed to do is they've sequenced the genomes of, I'm just going to paraphrase, it's big data, loads of TB from all around the world. And they've managed to grow it in these plates where they compared which TB would grow in the presence, which genomes would grow in the presence of which antibiotics. And from that, they've managed to work out from the 4 first-line antibiotics, by looking at the genome, you can tell which drug will work on it. And this is the first time they've ever been able to do this. So you can sequence the DNA of the TB from the patient, right, and you can know what drug will work on it. And this is a methodology, a protocol that could be applied in lots of different infectious diseases now, but they've done it first in TB. So I've got all the data from that, and I'm making an installation, working with Alex May, making an installation on that, about that at the moment, which is going to be premiered in a kind of lockdown way at the Ever Art Festival, which is usually in Moscow, this year online. I'm in a show called, I think it's called Neural Network Apophenia. So it's to do with looking at neural networks and big data and pattern recognition. So it's in this show, and it's going to be like this weekend festival in June. Okay. Anyway.
Yeah, amazing. Anna, listen, we could listen to you talk all night, but unfortunately I think we are out of time. So yeah, before I hand over to Autumn for just closing remarks, Anna, is there anything, any particular virtual events or exhibitions where your work is being exhibited? Is there anything you'd like to plug for yourself before we say our goodbyes?
There's a virtual exhibition called All in It Together.
Okay.
On at the moment, which you can see mentioned on my website, which is annadumitriu.co.uk. I've got an actual real life show on at the moment. You can go into a gallery and see some work, which is a piece called Archaea Post Singularity, Post Climate Change Lifeform, is in Kunstpalais Erlangen in Germany. And then I think the next show that I'm in, The Plague Dress, will be in July, hopefully. The aim is to open on July the 16th, I think, which will be at the Rijksmuseum Boerhaave in the Netherlands, which is going to be on for quite a while, the show, so over 6 months, I think. So, or at least, yeah, about 6 months. So I highly recommend it if you get a chance, you're in the Netherlands. If you're in Amsterdam, it's really easy to get to Leiden to go to the Boerhaave Museum. And if you go there, you can see the original Van Leeuwenhoek microscope.
Amazing.
So it's well worth a trip. So it's going to be in the, and I have to say that gallery is a former pest house where they treat patients for plague. So it's the perfect location in many ways.
Amazing. And is it just before we wrap up, I will say to everyone, to all the attendees, we'll of course follow up with an email and share the various links to the various different things we've talked about. I know we've talked about some of Anna's articles and works, and we've it's been a lot of people even putting their suggestions into the chat. So we'll send follow-up emails to everybody with all this information. So yeah, I'd like to say thank you, and I'll just hand over to Autumn now to close our session. But thank you so much, Anna.
Thank you so much, Anna. And yeah, as Amelia said, we've got our A-side email that goes out with our kind of leading articles to kick us off for every reading group. But then following up, we'll always come back at you with our B-sides if you want to take a deep dive into the different readings that we've got going on the theme. So thank you again, Anna. Thank you, everybody, for coming and joining us. From our quarantine caves to yours, stay safe, stay curious. And next time, we'll be talking with Claire Moriarty, Dr. Claire Moriarty, excuse me, philosopher extraordinaire, and we'll be talking about Euclidean geometry and a man with a very explosive intellect. Blew his hand off with dynamite. Come join us to learn more. Anyway, stay safe, stay curious, and we'll see you.